Hello students, how are you? Hope you all will be fine. Let's start today's lesson. It's all about the cell and the cell organelles. Irrespective of their size and shape, all life forms are made up of units called cells. The functions performed by the living organisms are also performed at the cell level. So cell is the basic unit of structure and function of all living organisms. In 1665, Robert Hooke discovered cell when he examined a thin slice of cord tissues under a compound microscope. He observed cells as empty chambers with thick outer coverings. However, the quality of microscope lenses improved greatly in the 19th century, which led to the discovery of cell nucleus in 1831 and many cytoplasmic organelles in coming years. The cell wall surrounds the plasma membrane of plant cell. It is rigid, inert, covering secreted and deposited outside the cell membrane. It consists of three layers, namely middle lamella, primary wall, and secondary wall. Algae have cellulose in their cell wall. Fungal cell wall is made up of chitin. Prokaryotes also possess cell wall made up of peptidoglycan. Cell wall is absent in animal-like protists. Middle lamella is made up of magnesium and calcium salts of pectin. It is sticky in nature that holds the neighboring cell walls together. Primary wall contains cellulose fibers arranged in a crisscross fashion. It is thin and flexible. Some plant cells, like xylem vessels, form secondary wall inside the primary wall. It is very thick and rigid structure due to the presence of lignin, which cements the cellulose fibers together. Cell wall bears tiny pores through which neighboring cells form cytoplasmic connections called plasmodesmata. Cell wall supports the structure of individual cells and the plant as a whole. It protects and gives shapes to the plant. Plant cell can develop turgor pressure due to the presence of cell wall. Cell membrane is thin sheet-like covering of the cell. Chemically, it is composed of proteins 60 to 80%, phospholipids 20 to 40%, and traces of carbohydrates. The structure of cell membrane is explained according to the fluid mosaic model. It postulates that cell membrane consists of double layer of phospholipids in which proteins are incorporated in a mosaic fashion. Cell membranes of eukaryotes also contain cholesterol. It prevents stiffening of cell membrane. Cholesterol is required for the fusion of secretory vesicles with membranes. Carbohydrates are either linked with proteins or lipids. Cell membrane acts as barrier and gatekeeper for the cell. It is semi-permeable, so some molecules can move across the lipid bilayer, but others are blocked. It maintains fixed environment inside the cell. Cell membranes act as a barrier between the cell and its environment. It regulates the exchange of materials between cell and its environment. Between the cell membrane and nucleus of the cell is an aqueous substance called cytoplasm. It is about 90% water, having many dissolved and suspended materials. It is the site for many biochemical processes. It stores food granules and waste materials. It is home for a variety of cell organelles which are discussed later in this video. Endoplasmic reticulum is a system of membranes present throughout the cytoplasm of eukaryotic cell. Flattened sacs of endoplasmic reticulum are called cisternae, which forms a network of interconnected channels. There are two forms of endoplasmic reticulum. Rough endoplasmic reticulum are covered with ribosomes. If ribosomes are absent, it is called smooth endoplasmic reticulum. A complex network of endoplasmic reticulum provides mechanical support to the cell. They are also involved in the transport of substances within the cell. Due to attached ribosomes, rough endoplasmic reticulum have role in the synthesis of some proteins, while smooth endoplasmic reticulum synthesize lipids including steroids. They also detoxify harmful substances. In muscle cells, smooth endoplasmic reticulum have important role in contraction process. Proteins make up to about 55% dry weight of a cell. A cell thus needs protein synthesis at high rate. This role is performed by the ribosomes. Ribosomes are tiny granular structures found both in eukaryotic and prokaryotic cells. They are not bound by any membrane. They are composed of roughly equal amount of proteins and ribosomal RNA. The prokaryotic ribosomes, however, are smaller in size. A large number of ribosomes are scattered in the cytoplasm. In eukaryotes, many ribosomes are also attached on the surface of rough endoplasmic reticulum. Each ribosome consists of two subunits, one small and one large. These two subunits join when ribosome has to perform its function. Golgi apparatus was discovered by Camillo Golgi. It is present in all eukaryotic cells. Like endoplasmic reticulum, Golgi apparatus is also a collection of flattened sacs like cisternae. However, in Golgi apparatus, many cisternae are stacked over each other. They are constantly formed at one end and break up into vesicles at the other end. Golgi operators store and modify materials into finished form before packing into the vesicles. Some of these vesicles settle in cytoplasm as organelles like lysosomes. Others fuse with the cell membrane to release out packed material as cell secretions. Products of glands like enzymes, hormones, mucus, etc. are secreted in cellulose fibers which arrange themselves to form cell wall. Lysosomes are single membrane bound small sac like structures. They contain a variety of digestive enzymes. The enzymes contained in lysosomes are synthesized on rough endoplasmic reticulum and then transported to Golgi apparatus. Lysosomes then bud off from Golgi apparatus with their possessed enzymes. One important role of lysosome is intracellular digestion. In this process, lysosomes digest material taken up by the cell from outside as food vacuole. When lysosomes fuses with the food vacuole, the lysosomal enzyme act on complex food substances and convert them into simple form. They also engulf and digest unwanted cell organelles. This process is termed as autophagy. Energy is an important theme in biology. All systems, from cells to ecosystems, require energy to work. Cells get energy by the breakdown of organic food in a process called respiration. If it requires oxygen, it is called aerobic respiration. It takes place in mitochondria. Mitochondria are found in all aerobic eukaryotic cells. Mitochondria are double membrane bound structures. The outer membrane is smooth and inner membrane forms finger like projections called cristae. They increase the surface area for the respiration. The fluid inside the mitochondria is called matrix. Mitochondria have their own DNA and ribosomes. They can multiply within the cell at their own. They produce energy in the form of ATP. That is why they are called as powerhouse of the cell. A vacuole is a membrane-bound fluid-filled sac. Animal cells may have many small vacuoles which exist temporarily. They contain water and food substances. Some freshwater organisms like amoeba and sponges have contractile vacuoles which collect and pump out extra water and other waste. Some cells ingest food by forming food vacuoles, which is then digested into simple molecules. Food vacuoles also store food. Plant cells have a large central vacuole. It is formed by joining small vacuoles. The membrane of plant vacuole is called tonoplast. It contains liquid called cell sap. Cells have had dissolved materials like mineral salts, sugars, and amino acids. It also provides support and helps in growth. The primary role of the central vacuole in a plant cell is to maintain turgor pressure within the plant cell. Turgor pressure occurs when the fluid content of a cell pushes the cell membrane against the cell wall in order to provide shape to the plant cell. Centrioles are hollow, open-ended, cylinder-like structures. They are found in animal cells. They exist in pairs near the nuclear envelope. Each centriole consists of nine triplets of microtubules. At the start of the cell division, centrioles duplicate and two pairs move to the opposite poles, thus help in the formation of spindle apparatus. They are also involved in the formation of cilia and flagella. Cell has a system of variety of fibrous proteins throughout the cytoplasm. These proteins collectively form cytoskeleton. Three types of cytoskeleton fibers are identified in the cell. These include microtubules, microfilaments, and intermediate filaments. Microtubules are made up of tubulin proteins. They are unbranched, hollow tube-like structures. Microtubules give rise to spindle fibers, cilia, and flagella. Microfilaments are very thin protein fibers. They consist of contractile proteins, mainly actin. They are responsible for streaming movement of the cytoplasm. The overall cell movement is also regulated by the microfilaments. Intermediate filaments are composed of a variety of protein including keratin and vimentin. They form a branching network in the cell. They maintain the cell structure. In tissues, they fix cells with each other. Cell activities like metabolism, growth, and reproduction need to be well regulated. In eukaryotic cells, this role is served by the nucleus. Nucleus acts as a control center of the cell because it contains hereditary material DNA. Nucleus is surrounded by two membranes which collectively form the nuclear envelope. Nuclear envelope bears nuclear pores at points where both membranes fuse with each other. Through nuclear pores, nucleus communicates with the cytoplasm. Some nutrients and proteins enter the nucleus through these pores and ribosomes and messenger RNA leaves the nucleus. Nucleus contains a fluid called nucleoplasm. Nucleolus is a round, darkly stained area in the nucleus. Ribosomes are assembled at this point. Here, ribosomal RNA is formed, which combines with protein to form ribosomes. It disappears for some time during cell division. Hereditary material in the nucleus is actually in the form of chromatin. Chromatin consists of DNA fibers coiled on histone proteins. During cell division, chromatin fibers condense into more tightly coiled threads known as chromosomes. Each species has its own unique chromosomal set different from other species. Plastids are double membrane bound organelles. They are found in plants and algae. There are three types of plastid, that is chloroplast, chromoplast, and leucoplast. Chloroplasts are usually oval in structure. Two membranes of the chloroplast form chloroplast envelope. They have their own DNA and ribosomes. They can multiply within the cell at their own. They have a system of membranes containing chlorophylls and other photosynthetic pigments. This system consists of hollow coin-like membranous structure called thylakoid. Many thylakoids stack to form granum. Some thylakoids of adjacent grana fuse to form intergrana. The fluid part of the chloroplast is called stroma. Chloroplasts synthesize food by photosynthesis process. It takes place in two phases. Light-dependent phase, which takes place in thylakoid membrane, light-independent phase, which takes place in stroma. Chromoplasts are colored other than green. They may be red, pink, yellow, blue, purple, etc. They are found in flower petals to attract insects. Insects help in pollination. They are also present in the wall of ripened fruits where they attract birds and other animals which help in seed dispersal. Leucoplasts are non-pigmented plastids. They are food-storing organelles usually found in roots, bulbs, and stem tubers. They store carbohydrates, proteins, or lipids. Some eukaryotic cells have extensions that look somewhat like hair. These structures are called as cilia. Some cells have whip-like extensions called flagella. Cilia and flagella consist of 9 pairs of microtubules which surround a single central pair of microtubules. Cilia and flagella are connected to the basal body. The basal body serves to anchor a cilium or flagellum to the cell. The function of cilia and flagella is movement. Some plant cell advantages and disadvantages are due to cell wall, adjoining plant cells are cemented with each other. Supportive structure of plant as a whole is thus formed by cell wall. Transport channels in plants, xylem and phloem, are also formed because of the presence of cell wall. The rigid wall helps plant cells to withstand high osmotic stress and store water. Plant cell can become turgid, which allows plant parts to maintain structure and stay upright. Plants cannot move from place to place because of rigidness provided by the cell wall. Due to rigid structure, plant cell cannot reproduce at faster rate. Some advantages and disadvantages of animal cell. The supportive structure of an animal cell as a whole is not dependent on the cell wall, but rather on the collective arrangement and organization of tissues, organs, and skeletal system present in the animal's body. In animal cells, since they lack a cell wall, the transport of fluids, nutrients, and gases occur through different structures and mechanisms. Animal cells cannot withstand high osmotic pressure and cannot store larger volumes of water. Animal cells cannot become turgid to provide support to the body, lack cell walls, which makes them very flexible. Animal cells can move. Animals can move to suitable environmental conditions, find shelter and better feeding fields and opportunities for reproduction. It also helps animal cells to divide and reproduce at faster rate. Thanks for watching this video. Like the video and subscribe the channel for more interesting videos.